Process chromatography, purification of chemical compounds. Title: Process chromatography, purification of chemical compounds. Introduction: 100 words. Process chromatography is an essential technique used in the purification of diverse chemical compounds. It plays a pivotal role in various industries, including pharmaceuticals, biotechnology, food, and beverage, and environmental analysis. By employing different stationary phases and mobile phases, process chromatography separates complex mixtures of compounds into their individual components. This article aims to delve into the fundamental principles of process chromatography, the different techniques employed, and its applications across industries. Understanding the intricacies of this powerful technique is paramount for researchers and engineers striving to optimize the purification of chemical compounds. Overview of process chromatography, 200 words. Process chromatography involves the separation and purification of chemical compounds based on their physicochemical properties. The technique exploits the differences in interactions between the tar get compounds and a stationary phase. The stationary phase typically comprises beads or resins packed into a column. When the sample mixture is passed through the column, the compounds interact with the stationary phase based on their affinity, size, polarity, or charge. This interaction leads to the separation of the compounds, enabling their subsequent isolation and purification. 1. Adsorption chromatography. Adsorption chromatography separates compounds based on their affinity for the stationary phase. The sample molecules adhere to the surface of the solid support, while impurities pass through the column. More quickly. By manipulating the composition of the mobile phase, the compounds of interest can be eluted, allowing for their further analysis or use. 2. Ion exchange chromatography. Ion exchange chromatography separates compounds based on their charge. The stationary phase contains charged groups that attract or repel the analytes of interest. By adjusting the pH or ionic strength (TH) of the mobile phase, the compounds can be selectively eluted, enabling effective purification. 3. Size exclusion chromatography. Size exclusion chromatography separates compounds based on their size or molecular weight. The stationary phase contains porous particles, allowing smaller molecules to enter the pores and be retained. Longer, while larger molecules elute first. This technique is particularly useful for separating proteins, polymers, and other macromolecules. 4. Affinity chromatography. Affinity chromatography exploits the specific interactions between the target compounds and ligands immobilized on the stationary phase. This technique can selectively capture and purify proteins, enzymes, antibodies, and other biomolecules by leveraging their affinity for specific ligands. Applications of process chromatography, 400 words. Process chromatography finds extensive applications in various industries due to its ability to purify and separate chemical compounds efficiently. Some notable applications include: 1. Pharmaceutical industry. Process chromatography is crucial for the purification of active pharmaceutical ingredients, APIs, and the separation of impurities. It aids in removing unwanted compounds, such as degradation product. TS, from the drug formulation. Furthermore, process chromatography plays an essential role in the production of biologics, such as monoclonal antibodies, recombinant proteins, and vaccines. 2. Biotechnology. In biotechnology, process chromatography is employed for the recovery and purification of biomolecules, including enzymes, growth factors, and nucleic acids. It ensures the production of high quality biopharmaceuticals and facilitates downstream processing in bioprocesses. 3. Food and beverage industry. Process chromatography contributes to the purification and analysis of food and beverage components. For instance, it enables the separation of sugars, amino acids, and vitamins from complex matrices. This technique is valuable in ensuring food safety, quality control, and the development of innovative food products. 4. Environmental analysis. Process chromatography plays a pivotal role in environmental analysis by separating and quantifying pollutants, toxins, and other contaminants. It aids in monitoring water quality, air pollution, and soil contamination, facilitating environmental stewardship and compliance with regulatory standards. Industrial applications of process chromatography are numerous and invaluable. The pharmaceutical industry heavily relies on process chromatography for the purification of active pharmaceutical ingredients, APIs, and the removal of impurities from drug formulations. It is an essential step in the production of biologics, including monoclonal antibodies, recombinant proteins, and vaccines. Biotechnology also benefits significantly from process chromatography. It aids in the recovery and purification of biomolecules such as enzymes, growth factors, and nucleic acids. By ensuring the production of high-quality biopharmaceuticals, process chromatography plays a critical role in downstream processing in bioprocesses. In the food and beverage industry, process chromatography is vital for the purification and analysis of components. It enables the separation of sugars, amino acids, and vitamins from complex matrices. This technique ensures that food safety standards are met, facilitates quality control, and supports the development of innovative food products. Furthermore, process chromatography has significant implications in environmental analysis. It is used to separate and quantify pollutants, toxins, and other contaminants in order to monitor water quality, air pollution, and soil contamination. By facilitating environmental stewardship and compliance with regulatory standards, process chromatography contributes to a safer and healthier environment. Technical advancements have led to the widespread use of process chromatography in diverse fields. The technique exploits the physicochemical properties of compounds to separate and purify them. Different chromatography techniques are employed based on the desired separation mechanism. One commonly used technique is adsorption chromatography, where compounds are separated based on their affinity for the stationary phase. This method is useful for purifying compounds with similar structures or properties. Ion exchange chromatography, on the other hand, separates compounds based on their charge. By adjusting the pH or ionic strength of the mobile phase, targeted compounds can be selectively eluted. Size exclusion chromatography is ideal for the separation of compounds based on their size or molecular weight. It utilizes porous particles to retain larger molecules while allowing smaller ones to elute earlier. This technique is widely used for separating proteins, polymers, and other macromolecules. Affinity chromatography takes advantage of specific interactions between the target compounds and immobilized ligands on the stationary phase. It is a powerful tool for isolating proteins, antibodies, and other biomolecules with high selectivity and purity. The applications of process chromatography are vast and extend beyond the industries mentioned earlier. It contributes to the purification of nutraceuticals, natural products, and fine chemicals. Process chromatography also plays a role in the synthesis of chiral compounds, where enantiomeric separation is crucial. Furthermore, process chromatography finds applications in forensics, particularly in drug analysis and toxicology. It aids in the separation and identification of compounds present in complex matrix, such as blood, urine, and tissue samples. Technical advancements have led to the widespread use of process chromatography in diverse fields. The technique exploits the physicochemical properties of compounds to separate and purify them. Diff.